morning and welcome to Marker Therapeutics webcast to discuss the Phase 1 clinical update from the Apollo study MT601 in relapsed lymphoma. At this time, all participants are in listen-only mode. Following prepared remarks, the webcast will include a Q&A session. You may submit questions at any time using the Q&A function on your screen. Please note this call is being recorded and will be available for replay on the company's website following today's event. I'd now like to turn the call over to Maria Bernadette Medel, Director of Corporate Operations for Marker Therapeutics. Maria, please go ahead.
Thank you and welcome everyone to today's webcast. Before we begin, we would like to remind everyone that today's webcast will include forward-looking statements. These statements are based on current expectations and are subject to risks and uncertainties that may cause actual results to differ materially. We encourage you to review the cautionary statements and risk factors described in our most recent SEC filings. We will now pause for a moment to allow you to read our forward-looking statement slide. Thank you. With that, I'm pleased to introduce Dr. Juan Vera, President and CEO of Marker Therapeutics, as well as co-founder of the company and one of the developers of the technology. Dr. Vera will provide an overview of the data from the company's Phase 1 Apollo study of MT601 in relapsed lymphoma, which was announced in a press release earlier this morning. We are also happy to have Dr. Jeffrey Schaus joining us today. Dr. Schaus is Assistant Professor at the Department of Hematology and Hematopoietic Cell Transplantation at City of Hope, Duarte, California, and he is one of the PIs in Marker's Phase 1 Apollo study. Dr. Schaus will share his perspective on the study and the broader competitive landscape. Before we open the floor to your questions, we will return to Dr. Vera for final comments. As a reminder, questions can be submitted at any time throughout the session using the chat box. With that, it's my pleasure to hand things over to Dr. Vera.
Thank you, Maria, and thank you everyone for listening. We're really excited to be here today to highlight our progress in our clinical study in MT601 in patients with lymphoma. Marker Therapeutics is advancing the MAR-T-cell technology, a technology that relies on millions of years of immunological evolution to provide a product that is capable of recognizing hundreds of epitopes present within the tumor, and in this manner, providing a treatment that can avoid the heterogeneous makeup present within cancer cells. This technology was developed at Baylor College of Medicine, where we have generated clinical proof of concept. And in addition to that, the company has been sponsored by non-diluted funding, including NIH, FDA, and CPRIT. And today, we're going to be providing a clinical update or MT601 in patients with lymphoma into our Apollo clinical study. But before we do that, let me just walk you through the MAR-T-cell technology and the mechanism of action. Next slide, please. Next slide. Thank you. So if we look at this representation, we see that the MAR-T-cell product is generated from peripheral blood T cells, which are isolated, assimilated with overlapping peptides with the purpose of enriching for tumor-reactive T cells, and in this manner, providing patients a product that is capable of recognizing multiple targets present within the cancer cells. And importantly, we're able to do so without the need for genetic modification or genetic enhancement. So in this manner, this is a natural product that we have now tested in the clinic with now early evidence of clinical activity and an excellent safety profile. Next slide, please. This is a quick overview into our manufacturing process. We're really excited now to start a strategic collaboration with Cellipont, a CDMO located in Houston, Texas, a vendor that will be responsible for the manufacture of this product in our future pivotal study as well as commercial launch. Now, as illustrated in this schematic, this is an autologous process, which our current vein-to-vein time is about 20 to 25 days. The final drug product is collected, cryopreserved, and sent to the clinical site in a cryo manner where it can be then administered. Next slide, please. This is a representation of our current pipeline. And as illustrated here, our lead asset is MT601, And today, we're going to review those clinical results. Next slide, please. MT601 is a clinical study that we're really excited and is now developing a MART technology which recognizes 6 different tumor-associated targets. And the results that we're going to be reporting today are results from a multi-center clinical study across the U.S. Next slide, please. The reason we are excited about these results is, one, the first objective is to recapitulate in a multi-center clinical setting observations from academic study conducted at Baylor. This study was published in the Journal of Clinical Oncology, where we were able to demonstrate the clinical activity of this technology in patients with lymphoma. Now, as part of the Apollo study, our objective is first to recapitulate these results in a multi-clinical trial setting, and two, see if we can even improve upon these results, as now we have performed certain enhancements to the process as highlighted in the right-hand side. More antigens, more dose, and a simplified manufacturing process that can yield a product that is 4 times more potent. Next slide, please. We can see here that this is the clinical study design, and let me walk you through the structure. This clinical study has 2 components, a dose escalation followed by a dose expansion. As shown here, the dose escalation range from 100 million to 400 million cells. And as shown on the right-hand side, this is open to patients with different types of histopathologies, including the LBCL. Today, we're going to show data on 24 of these patients. Importantly, as shown on the bottom of this slide, since June 17, the SRC has deemed the dose to be safe, and as a consequence, we're now going into our highest dose level, ever exploring humans, which is 400 million cells, as our dose expansion in patients with DLBCL CAR relapse. And this is important as this will be the foundation of our future pivotal study. Next slide, please. So these are the results that we have today. So we're really excited to report a CR rate of 50% and an overall response rate of 66% in our patients with non-Hodgkin lymphoma, patients that are heavily pretreated, and for many of which there's no treatment options. So we consider this to be really remarkable in terms of the clinical benefit that it can provide to patients. If we go then to the next slide and we examine the durability of responses, we see that of these patients, 3 of these patients have been in complete response for more than a year, and 5 patients have received the benefit of clinical responses for more than 6 months. If we go to the next slide and looking into this data in more detail, we see that many of these patients have different types of histopathologies. Many of them are DLBCL. They have received CAR T cells as well as bispecific. And despite failing to these prior lines of treatment, we are observing that MT601 is capable of providing complete responses which are durable. Now, if you pay attention in the left-hand side, I will point out that these patients have received clinical benefit despite receiving the lowest cell dose exploring this study today, which is 100 to 200 million cells. And from this perspective, I'm really excited and believe that these results we're going to be able to recapitulate, if not improve, as now we move into our highest dose level of 400 million cells. Now, if we go into the next slide, we can see that the highest dose level ever tested in humans, which is 400 million cells, have shown not only to be safe, but we also have seen objective responses. This is 9 patients, Hodgkin lymphoma patients, that have received the highest dose level of 400 million cells, where we're seeing an overall response rate of 78% and a complete response rate of 11%. And these are clinically meaningful observations. As illustrated here on the right-hand side, this is actually a patient with Hodgkin lymphoma from Dol Hakim, which is one of our PIs from the study from Kansas. This patient had a partial response, but despite this being classified as a partial response, you can see that this patient had high level of disease burden and it was controlled after the administration with MT-601. Next slide, please. And perhaps one of the elements that makes this technology very different to other alternatives is the safety profile. And what we have seen today is, as we explored 200 and the highest dose level for 100 million cells, we have seen an excellent safety profile without DLTs, no ICANS, and the only CRS has been reported has been level 1 in 2 patients that have been just really fever and response in absence of additional treatments. In addition to that, we continue to monitor the long-term toxicities, but again, remember, this is a natural T-cell product that does not require genetic modification, and as a consequence is not really subject to some of the long-term cell transformation that has been observed in cell therapy space using CAR T-cells. So with that adult, we're really excited to report today these clinical observations showing the efficacy of our technology in patients that are heavily pretreated, as well as confirming an excellent safety profile, even as we go now to our highest dose level. With that, let me hand over to Maria. Maria.
Thank you, Dr. Vera, for the insightful presentation. That was really a great overview of the data and what it could mean going forward. To build on that, we are happy to have Dr. Schultz with us today, who has been directly involved in the study as principal investigator in City of Hope. Welcome, Dr. Schultz, and thanks for joining us today. We're looking forward to hearing your perspective on the trial and how Marcus finding compare within the current treatment landscape.
Thank you. Thanks for the opportunity to speak about some of the exciting results that we've seen on this study. So I'll start out by going through the results on several of the patients that I've enrolled with large cell lymphoma. The first one, shown here, was refractory to 3 prior lines of therapy, including CAR T-cell therapy. And with MT-601, you can see at the 8-week time point that their disease it's gone into remission. Importantly, the patient had no toxicity associated with the treatment. On this next slide, you can see another patient, this one going through 8 prior lines of therapy, including CAR T-cell therapy. Again, showing at 8 weeks, complete metabolic response was achieved. And again, minimal toxicity seen in this patient, no CRS, no neurologic toxicity. This patient, shown on this next slide, achieved complete metabolic remission with prior bulky involvement of the spleen by 4 weeks after MT-601 treatment. And this patient had both prior bispecific antibody and prior CAR T, and a total of 10 prior lines of therapy. This last patient had cutaneous only relapse of DLBCL, was initially treated without lymphodepletion, had a partial response, then was retreated with remaining cells with lymphodepletion, and ended up achieving a complete metabolic response, again with minimal toxicity. So seeing this promising preliminary efficacy and this, in my opinion, incredible safety profile, we can dive in and see what the treatment landscape in CAR relapsed DLBCL looks like. And on this next slide, you can see unmet needs in DLBCL persist. So the treatment landscape has seen dramatic changes and continues to undergo changes associated with incorporation of novel therapies, including cell-based therapies like CAR T and bispecific antibodies. Even with these changes, however, unmet needs persist, including patients that relapse after CAR T or patients who are ineligible for CAR T due to toxicities. MT-601 has demonstrated exceptional safety, and we've seen the preliminary efficacy, and it carries the potential to fill many of these unmet needs. On the next slide, we've outlined some of the basics for the current treatment landscape in CAR relapsed DLBCL. And in the upper left, you can see bispecific antibodies are likely the best available treatment we have that's FDA approved in this setting. However, long-term follow-up suggests that the durability of response is not completely clear, and it's unclear whether these are curative therapies or just treatments that can buy patients more time. Below that, we see a combination of tafasitamab and lenalidomide, which itself has limitations in terms of efficacy and durability, and also in the pivotal study leading to its approval, high-risk patients were excluded from the study. In the upper right, antibody-drug conjugates such as polatuzumab and loncastuximab have shown efficacy. For pola, it's mainly been shown to be efficacious in combination with other treatments, and right now the most common use of pola is in the frontline setting as part of pola-R-CHP, and if it's being used in the frontline, its utility in subsequent lines of therapy comes into question. In addition, with lonca, although it has some efficacy in the CAR relapse setting, the number of patients included in the study that were CAR relapse was rather small, and the toxicity profile of loncastuximab is significant, with a significant proportion of patients on the study having SAEs. And then in the bottom right, we have other treatments like selinexor and the recent approval of BBR². However, these have not been widely adopted and have limitations in terms of their efficacy and toxicity as well. Looking to the future, there are many therapies currently in development. These include allogeneic CAR Ts, humanized CD19 CAR T with IL-18 expression, in vivo CAR Ts, other T-cell engagers, dual and tri-specific CAR Ts, CAR NKs, and other small molecules. The main limitation of these therapies that are in development is their limited scope. Many target one, possibly two antigens, up to three at times, but that leads to a limitation of antigen escape for a mechanism allowing for resistance and relapse. They also have short-term durability based on an initial study, and there may be increased toxicity as the number of targets are expanded in CAR T. In addition, the bispecific antibodies and T-cell engagers may end up moving earlier to earlier lines of therapy, and as such, there'll still be an unmet need in this setting. So taking this all together, on the next slide, we see that the current therapies have narrow recognition and short-term durability, and unlike T-cell engagers and CAR T-cells, the MT601 product does not depend on antigen expression for efficacy. And agnostic molecules like MT601 could address the current unmet medical needs in large cell lymphoma. So in summary, the last slide, MT601 is poised to fill the unmet needs in DLBCL. And despite the promise of the current novel therapies being evaluated, an unmet need persists, and MT601 has demonstrated, again, exceptional safety and preliminary efficacy and carries the potential to fill many of these unmet needs.
Thank you, Dr. Zhao, for sharing your valuable perspective and this brief overview of the treatment landscape in lymphoma. It's always important to hear actually the insights from those working directly with patients, and your comments really highlight the potential MT601 could hold for patients and how it could fit into today's evolving treatment landscape. Before we go into the Q&A session, we will hand it back to Dr. Vera for final comments.
Thank you, Maria, and thank you, Dr. Shah. I really appreciate your insight. Go to the next slide, please. I would just like to make some closing remarks. I think that definitely with our technology, we have something that it is quite unique and different, and that difference is precisely derived out of the mechanism of action. The ability of MT601 to target multiple antigens in a natural manner address perhaps one of the major obstacles of what I will consider as an effective immunotherapy. And I think Dr. Shah alluded to that, which is the restricted target profile that multiple of the current strategies are limited by. MT601 is able to recognize multiple antigens simultaneously, and in that manner addressing and overcoming one of the barriers which multiple technologies are currently limited by. We're really excited by the results we're seeing today. We thank all the participators in the study, the physicians, research coordinators, and definitely the patients. We will continue to work diligently to advance this clinical study with the objective of taking this product into the market. As illustrated in this slide, I think that this highlights a significant milestone as we have now finalized our dose escalation. We have observed no limiting toxicity. We have confirmed the early observations from the Baylor clinical studies. And now we're really excited moving into the dose expansion, which will concentrate enrollment in DLBCL, CAR relapse, and bispecific relapse patients, which will be the foundation of our future pivotal study. We look forward to continue communicating our progress and clinical outcome. And if you guys have any questions, please feel free to reach out to the company. We will be always available. Thank you very much.
Thank you, Dr. Vera. Thank you, Dr. Shao. At this time, we will begin the Q&A session of the webcast. So the first question, Dr. Vera, the Apollo study shows that 50% of the non-Hodgkin lymphoma patients have a complete response, with several patients maintaining responses more than 12 months, despite a median of 5 prior lines of treatment. How do you view this durability profile relative to CAR T cells, and where do most of the relapses occur within a year? So how would you compare the durability of MT-601 relative to CAR T cells as well as bispecific, which have shown shorter remission durations?
Sure, yes. Good question. Let me characterize the clinical landscape, right? And I think that these observations are considered to be remarkable, particularly given that we're seeing them in patients that are CAR relapse and some of them which are also bispecific relapse. And there is not a lot of literature in that regard when we look into the overall survival of these patients. However, from our best assessment, you basically see an overall survival for patients that are DLBCL CAR relapse, which is about 5 months, right? So without a doubt, the durability of responses that extend beyond 6 months to a year are very meaningful from a clinical perspective in relating to that particular clinical context, DLBCL CAR relapse, bispecific relapse patients. And also, as we just heard a moment ago from Dr. Shao, the clinical alternatives, despite many of the things that are characterized in the literature, are not that great when it comes down to a patient that is DLBCL and has failed CAR T cells. Right? I think that there's a lot of limitations in terms of what current treatments can bring to these patients in terms of the efficacy as well as the associated toxicity. So I think that the question is valid, right? Which is how do we characterize the results we're seeing today against the competitive landscape related to products that are approved, products that are coming into the market, and the overall survival of these patients. And I think that is quite significant given the fact that, again, you're talking about heavily pretreated patients that the current treatment options are not really effective, and you have to combine that with an excellent safety profile. I think that the combination makes this very exciting and powerful clinical observations.
Perfect. And CAR T cells are usually used with lymphodepletion. But in your study, you used with without lymphodepletion. How do you interpret this finding in future trial design?
Yeah, that's an excellent question. I think that we have seen objective responses from our technology even in the absence of lymphodepletion, right? And I think we briefly reported those observations in our past clinical release. The incorporation of lymphodepletion has been performed with the objective of influencing a positive manner the expansion and persistence of the T cells. This phenomenon has been, I think, recognized across multiple different adoptive treatment programs. And we have been able to recapitulate those observations. And a few months ago, we actually reported our findings, what we actually have seen in our data set when we compare patients with or without lymphodepletion, that the incorporation of lymphodepletion is able to influence expansion and persistence of the T cells from the patients. Now, the thing that is important to recognize is that despite the incorporation of lymphodepletion, we have not seen a detrimental effect in the safety profile. And again, one thing that from my perspective, I wanted to point out is lymphodepletion is not the problem. The problem is the side effects derived when lymphodepletion is combined with other side treatments such as CRS and long-term neutropenia and infections and so on and so on. And I think that from that context, the incorporation of lymphodepletion has not had those effects with our technology, right? And the importance of that is that has provided very clear guidance in terms of our direction going forward for our registration study. And going forward, the large database that is presented today includes patients that have received lymphodepletion, and going forward, patients will be conditioned to the standard lymphodepletion as a regimen for the administration of the cells.
Talking about the registrational study, maybe you can walk us through anticipated timeline for approval of MT-601 plus the market opportunity.
Yes, that's an excellent question. I think that this is something that is going to become our next critical milestone, which is advancing and contextualizing these results for a future approval. And to that end, I think what becomes most important is how this is a high unmet medical need, right? And I think that that is characterized by the limited options that many of these patients have, the high level of toxicities with the approved current treatments. And I think that when you take that in context with the result that we have seen today, then you have a very strong case to make in order to have this product advanced for accelerated approval, right? We still have to meet the FDA and present our proposal, but I believe that our clinical data have a strong support for that particular developmental plan. And in that context, I believe we'll be able to get the product approved with a single arm phase 3 clinical study. But again, the fine details in terms of the start of the clinical study still has to be discussed in an upcoming conversation with the agency.
Thank you. And what are your thoughts on MT601 eventually moving into earlier lines of treatment?
Yes, absolutely. I think that is definitely the future, right? We recognize the reality and we are very strategic when it comes down to deploying our product. And as a consequence, we are advancing MT601 in DLBCL CAR relapse patients with the objective of demonstrating in a patient population with an unmet medical need that without a doubt this treatment works. And that it does so while providing an excellent safety profile. And the reason to do that is so that we can get approval nevertheless with a restricted label, but we get approval and start changing the dynamics of the company and get to a point of revenue with the objective of soon after that start extending this to an early line of treatment. And I think that from what we have seen today based on our data, the Baylor data, I think that it's reasonable to assume that is a realistic future where we'll be able to move this treatment even before CAR T-cells.
So for the dose expansion, you mentioned that you will focus on patients with DLBCL who have relapsed after CAR T-cells, and you also mentioned the favorable safety profile of MT601. Would you also consider enrolling patients who relapse after bispecifics in the dose expansion cohort?
Yeah, that's an excellent question, and allow me then to clarify. And I think that the first thing to understand here is that when we look at DLBCL, that's a highly fluid clinical space, right? And I think that recently we have seen the approval of bispecifics. We also see bispecifics now moving into an early line of treatment. And what I'm trying to say is that it's at this point very difficult to predict the final evolution of the clinical landscape for DLBCL CAR relapse patients, right? But it is likely to assume that bispecifics will move potentially in a clinical application before CAR T-cells due to the fact that they're easier to administer given the fact that they're off the shelf. And as a consequence, what we're envisioning for the patient population is to treat patients that are DLBCL, CAR, and or bispecific relapse. And from that perspective, have a more inclusive criteria that will be able to encompass patients that will fail one or the other or both, given the fact that this is still an evolving clinical landscape.
Thank you. We received many questions, but I think we have time for one more case. So going back more to the scientific part, so given the strong efficacy that was seen with the lower doses after tested at 100 to 200 million cells, what is now the scientific and clinical rationale to move into the higher doses in the dose expansion in patients with DLBCL?
Yeah, that's an excellent question. And I think that the overall premise, which is very simple, is that more is better, right? And I think that there is a precedent for that viewpoint. I think that especially in the field, we have seen a correlation with other target drugs, including CAR T-cells, where there is an association as to increase the number of cells and the ability for cells to expand. There is an effective target measure that is able to provide more reliable and reproducible and objective responses. Right? So again, the overall viewpoint is that more T-cells will basically provide a higher effective target ratio and therefore deeper responses to patients. Now, I think that many different programs have a limitation in terms of pushing the boundary as you get to more cells. There also increases toxicities. I think that what we have here that is different is that despite going to the highest dose level, which is 400 million cells, we haven't seen anything detrimental from a safety perspective. So with that said, if you take a step back and look at those 2 points, we are advancing the expansion cohort, which is the DLBCL CAR relapse or bispecific relapse population with our highest dose level with the attempt of recapitulating but improving the clinical responses that we presented today, as we know we're going to be treating patients with more cells without necessarily affecting the excellent safety profile, which again makes this treatment very different. So again, we still have to collect that data, but I think that there is good scientifically rationale from our programs that suggest that more cells will be able to have deeper responses. But again, I'm really excited that now we move into the second part of the year and concentrate enrollment in patients that do have CAL and all that is relapse into the higher dose level, and I think that would be a very strong foundation as now we go into our future clinical study for next year.
Wonderful. Thank you very much for the overview and the nice summary. So we received a lot of questions, as I mentioned, and I would like to thank everyone for your questions and hope you addressed most of them. On behalf of Marker Therapeutics, we would like to thank Dr. Vera and Dr. Schultz and all of you, of course, for joining us today, and we appreciate your interest and engagement, and wish you a wonderful day.
This concludes the consult. You may now disconnect.